So I think today, to begin with, we're mostly done with meiosis for now. Even though you see the word meiotic drive in here, we're not going to be talking about chromosome pairing or recombination much, except I think we'll just review a little bit the last time because there'll be some things to review for the next quiz. But first I think we should, or I should go over what's going to happen for the rest of the class. So it's the middle of June, and we have another 1, 2, 3, 4, 5, 6. We're going to meet up 6 more times, and one time is going to be for the final test. So I thought this is how those last few classes are going to go. So if you remember the first day of class, I had this idea that maybe we would do some presentations, like we would grab a journal article and present that to the class. But this class hasn't really been that interactive with a lot of question and answering. It seems like for the main part, I get up here and blather for an hour and a half, and you guys are hopefully absorbing some information about chromosomes. So I think maybe we'll just keep going with that. Because there's some things that I wanted to cover as well that maybe it would be a good idea to just skip the presentation part. So I think we're gonna change the schedule a little bit, more a little bit like the original syllabus was. So there weren't any presentations on that to begin with. So you'll notice. Okay, so first of all, today we're going to be talking about the evolution of human chromosomes, especially, but also other animal chromosomes, and then this interesting phenomenon called meiotic drive, where we can see Mendel's laws being violated. Then the next 2 readings are going to be 2 chapters on sex chromosomes. So that's a quite important part of chromosome biology, and they show up in many different contexts. So human disease is quite important. Sex chromosomes are important there, and also in basic biology, how animals determine, and even plants, how they determine what male or female, what sex they're going to be. So we're going to break that up into 2 classes, but this June 27th, there's going to be a quiz first on this kind of second, third of the classes. Okay, so there's going to be everything that has been up until this point, so everything up until there could be on the quiz. And then there's going to be 2 more classes. This one, July 4th, is going to be about methods, mostly microscopy methods, but also some genomic and bioinformatic methods. So kind of a lot of computational stuff, both computational image analysis of chromosomes, as well as some introductory points to how people are studying chromosomes on the genome sequence level, so the whole human genome, or the whole yeast genome, things like that. And then on July 11th, the last little bit of class material will be the connection between chromosomes and cancer. Cancer has been called by reputable people to be a disease of the genome, or a disease of chromosomes. And chromosomes are really the focal point of what goes wrong during cancer. Chromosomes usually break up, rearrange, or some get lost, or some get extra pieces. So there's a really big important connection between chromosomes and cancer that we will go over for part of this class. And then the rest of that will be kind of going over everything we've learned in the class so far from day 1. So probably the first half hour or so I'll cover this, and then the last hour will be kind of going through a lightning round of the entire class up to that point. And then from my understanding of this schedule, you can correct me if I'm wrong, but it looks like Tuesday is kind of a special day because it's a week ahead of the other classes. So on July 18th, according to the calendar, it looks like we are not going to have a class unless I have to miss one of these for some reason and then reschedule it. It's like we have a built-in extra day. So we're not going to have any class that day, but then we'll meet again on the 25th for the final test. Okay. Any questions about that? No? Okay. So first, let's I thought the first thing we should do, since there was kind of a lot of tough material and some new vocabulary from last time, I'll just go over a little bit and ask for some people to volunteer information. So just I have a few slides from the last 2 lectures, and I just wanted to ask if anybody remembers. So to initiate recombination, you've got to cut DNA. And does anybody remember what is the enzyme that cuts DNA? What is it? Spoilin. Yeah, that's right. Yeah, SPO11. And then, do you remember how many SPO11 molecules are involved in this cutting process? Yeah, that's right. You get 2 SPO11 molecules that make that cut. That's right. Okay, that's how it works. And then the next step after you've cut and resected, this single strand comes in and invades this double strand, and then it makes this thing pop out. And does anybody remember what that thing that comes out is called? No, you remember? Yeah, that's right. Yeah, that's the D. Yeah. And it's shaped like a D. If you just turn it on its side, it's like the letter D. So it's D formation. Okay, and then that you get, after this D loop pops out, you get new DNA synthesis, and when the D loop finally touches the upper cut strand, now this new strand has a template that it can be repaired on. And then after some synthesis happens, and then so there's 2 things here shown here. One is the name for this structure as a whole, and then there's this region in between has some special name for it. So you remember what is this structure called? There's 2 of these things. Do you remember the name of this? Yeah, so it's so they're joined together, so it's a kind of junction. Junction is part of the word. Do you remember? Okay. I know you know what it is. Yeah. So these 2 things together, the whole thing is called a double holiday junction, and it's spelled like this: holiday. Even though the regular word for holiday only has 1 L, but this is somebody's name. This is the name of some guy, and he spells it with 2 L's, so it's a proper name, capital H, so that's why it's called double holiday junction. And then in between this, there's a special name for this region, which might contain DNA from 2 different molecules. Does anybody remember? The region between the 2 holiday junctions. So for instance, this D loop up here is unchanged from the bottom strand. So it's possible that this DNA is not complementary. This green DNA may not be complementary to the blue DNA. You may have an A across from a G instead of a T, for instance. So that kind of mismatched DNA, there's some name for that region. And it's a little bit tricky, and I wrote it really small for some reason, but it's heteroduplex region. So duplex, I guess, because there's information from 2 different strands, and hetero because the information might be different. Okay, so that's double holiday junction and the heteroduplex region. And then just to go over again really quickly, so this is another view of the holiday junction, double holiday junction, and you get either a crossover or non-crossover depending on which way you cut it. So if I were to cut them in parallel, so cut this one that way and this one that way, what would that lead to? If I cut here, so I cut this strand and that strand, and then also I cut this strand and that strand, what would happen?
Crossover.
Yeah, right. That's right. Yeah. So, yeah, that would lead to a crossover like that. And then if I cut the strands that way, of course, it would be non-crossover. So you can see just by, all you have to do is change one, the way one is cut. So if you hold one constant, then depending on which way you cut the other one, you get either a non-crossover or a crossover, and also the, yeah, right, so you can also see that reflected in the bottom. Okay, this slide, right, okay. All right, so that's just a brief overview of last time recombination. I might put some of that on the quiz. Okay. So for the rest of the time we have, I'm going to talk about chromosome evolution and then a related phenomenon called meiotic drive. So before we start talking about chromosome evolution, there's one bit of chromosome nomenclature, very faint font, that I should have introduced a while ago, but better late than never. When people talk about chromosome numbers, they often use these letters N and X to talk about chromosome numbers. And N usually refers, well, N always refers to what is called the haploid number, and that has a special meaning, which is just the number of chromosomes in the gamete. So you can think of it as the number of chromosome pairs as well. So humans have a haploid number of 23, but when we say how many chromosomes does a human have, we usually give a little more information, and we say 2n equals 46, which means not only do we have 23 pairs of chromosomes, but it also says we're a diploid organism that usually has, most of our cells are diploid cells. So if you were talking about a haploid organism, you might say their chromosome number is n equals 10, and that means they're usually haploid and they have 10 chromosomes, and they only double their chromosomes when they reproduce. So n just refers to that number. But there's another basic number of chromosomes you can think about, which is called X. And this is a special, the basic number is a special number that is for organisms that have multiple genomes in one cell. So a lot of plants, for example, and especially a lot of crop plants, a lot of plants that we eat, are made when two related species of plant come together and their genomes just mix together in one cell and they stay there. So they actually double their entire genome size. And wheat is a very important example of that. So there were all these in Turkey or Ukraine or somewhere over there, or Iraq, so some place where wheat was domesticated. I'm not really sure if it's known where. But all of these, I'll say 2 minus 14, all of these, they're just wild grasses growing around that had a diploid number of 14. But at some point, these guys got together and they became a new species. So usually species branch out. One species will become two species. But sometimes especially if they haven't been apart for very long, sometimes they can come back together. So two species came together into one, and then later it joined up with another one. And so what this organism now has 42 chromosomes, and this is what we know of as wheat. It's the normal kind of wheat that we use to make flour and bread and everything. So what happened was its genome tripled, but you can still see the original genomes in this organism. So this is how you would talk about its chromosome number. So you would say 6x is 2n is 42. So x, the basic number, is 7, which is the haploid number of these grasses. If you look at its chromosomes during meiosis, you can see the genomes are still kind of separate, doing their own thing. And chromosome 1 from this organism still pairs with just chromosome 1 from this organism. So they have 3 genomes all together living in the same cell. And there's many examples of that. I guess strawberries have 4 genomes, and kiwi fruit have something like 12 genomes or something like that. So a lot of fruits and vegetables and grains have multiple genomes inside them. That doesn't happen so much with animals, but it's good to know if you ever come across a description of some species and you see an X and an N that are different numbers, that means that organism has multiple genomes inside one cell.
Sorry.
Yeah.
So the gametes is diploid, like for those. How is it possible with the gametes?
No, so the gametes are still haploid. So N is still 21. So a single gamete, a single pollen from this wheat will have 21 chromosomes.
Yeah, but how is it possible if it's haploid, like because it's crossing with another species? How can it ever become 2N? Like,
Oh, how does it? Yeah, that's a good question. So what happens is if you have a pollen from one species goes and fertilizes the fruit of another species, and it goes in there. But because of some incompatibility or some mistake, instead of, I don't actually know the seed anatomy very well, but you've got a nucleus now with, so its original haploid number was 7. The 7 chromosomes came in, they met the 7 chromosomes of the other species, but instead of directly going into mitosis, so it replicates, and then there's this thing called endoreduplication, where first all the chromosomes double, and then come apart, but the nucleus doesn't divide, so it just bulks up.
That makes sense, but then it's not 2N, like in sense, because in humans it's not like we have the double, like we don't have 23 pairs of identical chromosomes, because it's come from father and mother, but now the 2N basically a double of N.
So it has to start that way. But I guess if you imagine these two plants are out there in the field, if it just happens once, you'll never get another species. But somehow this guy, this little grain with 2 identical genomes, grows up, and now its pollen has twice the number. So if that one now happens to land on another one like it, or it could self-fertilize too. So so eventually, it's more likely it's going to land on another plant that just happened to have that. It's a very rare event for it to happen, but I guess it's thought that because this happens so often in human-selected plants, crop plants, and fruits and things, the result of getting this is the seeds are bigger because they have twice the genome. So we think about people 10,000 years ago looking for a grass with bigger seeds. They'll spend all day looking at grass, and when they find one with big seeds, they'll keep it. So humans are out there finding these rare events and amplifying them. So it doesn't. You'd probably wait a long time and you'd never see it happen, but over thousands of years, eventually a rare event to get multiple. That's a good question. Nowadays, we can make that happen artificially by using drugs to. At the moment where the cells are about to divide, you dissolve the spindle, and the nucleus collapses, and then it thinks it went through a division, and it starts from there with twice the number of chromosomes. Okay, but so that was just a little digression into possibilities of multiplying your genome. For some reason, plants can do it really easily, but not too many animals have done it. I think a few cases of rodents exist, but in the primate lineage, this does not happen. So humans don't seem to have any kind of mixing of genomes. But that is not to say that humanoid chromosomes do not change. Okay, I just gave it away. But so we know that the N number for humans is 23, and I just showed you. I was going to have you guess, but interestingly, the number of chromosomes, so the haploid number, the N number of all the other great apes is 24 instead of 23. So it seems like there's some discrepancy, and I just want maybe can someone come up with a hypothesis as to what's going on? Why do we have 23 and gorillas and chimps and orangutans have 24? Anyone think of some possibilities as to how to explain this?
Non-destruction.
Yeah, so right, it's possible that there could have been a non-disjunction event. So of course, first of all, it's if these guys and these guys and these guys all have 24, it's very likely. So down here, there's another species that lived this long ago. That's the ancestor of all these species. So it likely had 24, right? Because that's the most parsimonious. That's the most the fewest number of changes have to happen in order to give rise to the modern numbers. So we assume this guy had 24 chromosomes. Then at some point, maybe one chromosome got lost in some non-disjunction event, right? So on the lineage somewhere leading to humans, so after this branch, maybe one of our ancestors lost a chromosome. But chromosomes usually have a lot of genes on them, and we know if we lose any of our chromosomes, it's almost always fatal, unless it's in one of the sex chromosomes. So although that could happen, it's unlikely that we would lose an entire pair of chromosomes. So any genes that were on that chromosome being gone probably is not gonna happen. The other option, apes gaining one chromosome, the other apes would be as if this was 23, this guy had 23, and then somehow independently on this branch, and this branch, there was a chromosome gain event. But the likelihood of those three different branches gaining one chromosome is not very high. So I'll tell you, that didn't happen either. So if we didn't lose a chromosome and they didn't gain a chromosome, are there any other options?
Could it kind of all of them just stuck together?
Yes. Yeah, that's the last option, and that's what happened. Okay. So these guys with 24 chromosomes, if two chromosomes came together and turned into one chromosome, then we wouldn't lose any genes, so that we'd be losing a chromosome number. We'd be losing one from our N, but that wouldn't actually make us lose any of the genes. And the way we know that is we can look at chromosome bands. Remember, we can stain chromosomes with different stains, and then either by the AT versus GC content, or by the density, or more likely both. Each chromosome will have a very unique banding pattern. And this is what human and gorilla and chimpanzee and orangutan chromosomes look like. This is human chromosome 3, along with the ones from chimps, gorillas, and orangutans side by side. So you can see they're quite similar, right? We can actually see these banding patterns, especially between humans and chimps. Our closest relatives are almost identical. And that's also the case for chromosomes 4 and 5. So we can use this to track what happened. And we can use these kind of banding patterns to look. This is kind of a diagram. And here, these are also different chromosomes, 1, 2, 3, 4, 5, and 6. This is showing the P arm, the petite arm, as I'm known. The P arm, they're all identical. And if you look at the Q arm, they're also identical. But this spacing in chromosome 2 may give you some clue as to what's going on here. Because when people did this spacing, they saw that the P arm of human chromosome 2 resembled one of the 8 chromosomes very strongly, and the Q arm of human chromosome 2 resembled a different A chromosome very strongly. Now you'll notice that the centromere of our chromosome 2 is in the exact same place, or a very similar place, to the chimpanzee chromosome 2's centromere. The other chromosome from apes that joined up with this one, it also has a centromere. And if you look in the human case, we have the exact same region, but now there's no more centromere there. So somehow these two chromosomes got together, but instead of having two centromeres, now there's only one centromere. Can anyone think of why that happened? Why did one of the centromeres go away? Why would that make sense? That to happen? Yeah. So having two centromeres, being dicentric, so two centromeres, is a very dangerous thing to be, because even though, so you might think, okay, there's no problem, you just pull both centromeres to the opposite spindle poles and you'll be fine. But there's actually no guarantee. These centromeres don't know what these centromeres are doing. These two centromeres may be actually reversed. So this isn't a crossover, this is just one chromosome being twisted. And if this centromere is pulled this way, and this centromere is pulled the other way, then this chromosome will just get ripped in half. So what likely happened is, yes, these chromosomes had some tendency to fuse after a while, and then they got ripped in half. But every once in a while, another rare event would happen, and these centromeres would get inactivated, and then that could propagate. And then that's what the actual chromosomes look like. Yeah, this was it was pretty easy just with this kind of primitive banding pattern, staining and counting the number of bands and measuring them, that people could figure out what happened. And this is the consensus now, is that somewhere along this lineage, so one of our human ancestors, we don't know which one, but somewhere after the split between chimps and humans, two ape chromosomes fused into chromosome 2. And if you look at other parts of chromosomes, so human chromosomes and chimpanzee chromosomes are not completely identical. So there's that big gross change of chromosomes fusing into chromosome 2. But if you look at other regions, you can see, okay, although we have pretty much the same genes, so the chimpanzee chromosome here is shown on top, and the human chromosome is shown on bottom, and there is this consensus between the two chromosomes. But if you look very closely, you can see some regions where the order of genes gets flipped around. So in humans, there's this gene RALA, and it comes next to CDK13. But in chimpanzees, that order is reversed. So at some point, a little tiny piece of the chromosome broke off, but then DNA repair came in and fixed it, but it fixed it by flipping it and putting it the other way. And if you think about chromosome structure, that's not too hard to understand. If this happened during when the chromosome is existing as some sort of loop, so if the break happens here, then it would be quite easy to imagine that it could get fixed this way, and then this loop would just be flipped around and everything on that one loop would be reversed and start facing the other way. So it's thought that perhaps these micro inversions also that you can see over evolution, they'll reflect something about the chromosome structure and the size of these loops that could possibly be inverted through DNA repair. And then the final kind of proof that this chromosome evolution has happened in the lineage leading to humans, the easiest way to see it is by using this fluorescence in situ hybridization, which, as you remember, you can call up a company and order some DNA that's been labeled with a fluorescent dye. And since we know the entire human genome, we can specify whatever sequence we want and have that DNA localized wherever in the genome we want. So we can label each chromosome with a different chromosome paint. So if we take the chromosome paint that normally labels human chromosome 2 and apply that to chromosomes from different apes, you can see quite well that human chromosome 2 has, of course, there's only 2 of them, but the same exact paint for chromosome 2 hits 4 chromosomes in orangutans, with the exception of the very tips of these chromosomes, which is probably lost during this fusion event. Okay, so like this genome doubling and tripling that's happened before, you've also got to think that this, how did it spread throughout the lineage, right? So, of course, you start off, there's a branch, and you can imagine, okay, the chromosome fused somewhere along this branch, but really it fused inside a single individual. One person, or one ape human, had this fusion event happen in their own chromosomes. But then they would need to mate with another ape man to pass this on. And then their offspring would have, they would be kind of heterozygous, right? They would have one modern chromosome 2, and then they would also have these other chromosomes, which I don't know the numbers for these 8 chromosomes because the apes have different numbering systems, but we can call them 2A and 2B. So the person who had the fusion has a kid with a person who doesn't have the fusion. And now in this kid's germ cells, they've got this heterozygous situation. And they have to do meiosis normally, which means they have to have crossing over between these 2 sets of chromosomes. And then, since these still have their original centromeres, there's the possibility that, again, you might have non-disjunction. You might, because you've got different centromeres, and this centromere doesn't know what this centromere is doing, it's possible that you will lose one of these chromosomes in the next generation. So you might think that having this fusion event would actually be a disadvantage in evolutionary time. So you would not expect to ever see chromosome changes. And that's quite true. And the fact is that this is 12 to 16 million years, and chromosomes only change by one in all that time. So it is extremely rare, but it still does happen from time to time. And if you look at hospital records, you can see all kinds of chromosome rearrangements. Everybody's likely to have at least one micro deletion or micro insertion in our own genomes. But for that to get fixed throughout the entire population will take a lot of time. However, there is there's one process or one phenomenon in cell division that can actually accelerate this process and lead to a rapid takeover of the entire population by a particular chromosome type. And that's this phenomenon called meiotic drive that I'm going to spend the rest of the time talking about here. So, let's run that. So if you think about over time, how these chromosome numbers might change, you start off with just one individual getting a break or a fusion, some difference in their chromosome number. So this is what their gametes would look like. So an individual with unbroken chromosomes would have to get together with an individual that has a broken chromosome or a fused chromosome. And eventually, as you look over many generations, eventually that has to get fixed in how this individual has 5 chromosomes, where before they only had 4 chromosomes. So how can that happen? There's actually three different mechanisms how an alteration in chromosome number might get fixed in the population, so that if you look at every individual has the same one. So these three different mechanisms are genetic drift, and then you could have actual selection, and then this third one called meiotic drive. Genetic drift is really just, it's not really a mechanism at all, it's just randomness. It's just based on the fact that, so in every generation, so I said that this configuration of chromosomes might be deleterious, and you have a high likelihood of having non-disjunction and losing one of these chromosomes. But of course, there's also a 50% chance that you won't, right? 50% chance that these chromosomes will go the right way, and this one will go the right way, and everything will be fine. So it's possible for events that even slightly low probability events like this, if you try enough times, either that this individual will eventually have a healthy offspring, and if there are enough of those healthy offspring and they mate and they get their alleles into the population, eventually, even though there may be a problem with this, this low probability events do have some chance of taking over the population. And this is just kind of a simulation of that happening, where each line in this simulation would be one generation in a population, and the white dots would be one individual that has some new mutation or a new gene like this. And then depending on how likely it is for that to get transmitted down to the next generation, sometimes you can see more individuals have it, and then at later times fewer individuals have it. But if just by random chance, for instance, let's say a volcano explodes or something, and everybody dies except for just a few people that have this weird configuration of chromosomes, then that's all there is left. And then the entire population would eventually just have this kind of, or no, this is fusion. So a volcano could kill everybody with that, and leave only the ones with fused chromosomes. So yeah, just by random inheritance, you can have some fluctuation that will eventually lead to the entire population, and there's just the last non-fused individual dying out at the end. And you can come up with over how many generations does it take to get complete fixation, so 100% of individuals have a certain genetic makeup. This is just some random thing that can happen over time. But of course, you're just as likely to, of course, kill everybody that has the new fusion and only keep the original people. In fact, if it's a mutation that causes some problem, you can see that there's more of the original people without the mutation than there are ones with the mutation. So genetic drift is a plausible mechanism for leading to for a new allele to become widespread throughout a population, but it requires some low probability events to happen. So it's not very common. It's not a very common thing. So another possibility, right, would be if one chromosome variant gets selected for. So this means that, for instance, let's kind of go in reverse, and let's say, what happens if one chromosome splits up into two chromosomes? Are there any cases where this would be an advantage to an organism, such that even though you might have different numbers of chromosomes for a little while, this configuration of having two smaller chromosomes might be better than this configuration with a big chromosome. And there is one case where just chromosome length itself could be selected for, and that's if the chromosome happens to be too long. So if you look at human chromosomes and other chromosomes from other organisms, You can see, so our chromosome 1 is the longest. Chromosome 2 is also pretty long. We have this sort of range of chromosomes down to the 20s that are very small. But what we don't have is another chromosome that's really, really super long, right? And if you look at the distribution of chromosomes, the longest few chromosomes are actually pretty close to each other in length. And all organisms are like this. So most organisms do not have like 1 or 2 chromosomes that are really, really super long. And the reason is thought to be that just the mechanical requirements of cell division put an upper limit on how long a chromosome can be. So if you have your chromosomes lined up the metaphase plate like this, let's say there's one chromosome that's really, really long. Okay, and then they have to segregate into opposite cells. So the spindle can pull them into opposite cells, but it can only pull so far. And if one chromosome is really, really, really long, you might end up pulling it all the way here, but it's still got part of its arm stuck in the other side. This can happen actually if you use irradiation or something and artificially fuse chromosomes together. You can end up with chromosomes that are too long to go through the cell division. And what happens usually is that the cell can't really detect this too well, and so they'll divide the cells anyway. And then oftentimes this chromosome might get caught in the middle and get chopped off in the end. So having a chromosome that is too long can actually get selected against. And over time, chromosomes can increase in length more and more as, for instance, transposons and viruses and other things can just invade chromosomes, and so they can get longer and longer. So at some point, it may make sense for the chromosome to split in half, and that can be selected for by this configuration of having 2 split chromosomes can then spread throughout the population. So this mechanism probably kicks in long before the chromosomes get really, really too long, because even losing a little bit of chromosome in cell division would already be selected against. So anyway, this is another way that chromosome fusion or fission could be selected for or against. But other than those 2 mechanisms, so there's random chance and there's actual selection based on fitness, based on how good it is for the cell to have this configuration. There's this third mechanism called meiotic drive, and that is that certain chromosome configuration can become common, can spread through a population by exploiting this kind of loophole in the machinery of meiosis itself, regardless of the effect on what the fitness for the organism is going to be. And so what am I talking about the machinery of meiosis and how it can be exploited? So the conditions that can give rise to meiotic drive are first, you need some kind of asymmetry in the products of meiosis. So, meiosis gives, you have meiosis 1 that gives rise to 2 cells. And then meiosis 2, now you have 4 cells. If those 4 cells are all identical, then it doesn't matter which cell a chromosome ends up in because they're all identical. But if these meiotic products are different, then it really matters which one the chromosome gets into. I'm going to actually skip to the example and then come back to these conditions later because I have to go over this kind of cell biology. So in male meiosis, right, this is meiosis 1 and then this is meiosis 2, and you end up with 4 sperm that are pretty much all identical to each other. So every spermatocyte that enters meiosis is going to give rise to, if everything goes well, it's going to give rise to 4 identical spermatids. But in female meiosis, right, there's a difference here, and that is the first meiosis gives rise to this asymmetric, there's this asymmetric division where you have the oocyte and then this polar body. So that's something to write down. So I think that's called kyokutai, this little. So the first meiosis is asymmetrical, and then also so is the second meiosis. So the polar body rarely, if ever, actually divides. It usually just sits out here and degenerates. So at meiosis 2, in female meiosis, you're left with only one viable meiotic product. Only one cell will live on for the next generation, whereas in male meiosis, all 4 of them will live. So if you are a chromosome or a pair of chromosomes that have split, it really matters whether you end up in here or whether you end up in here. So for instance, if you are this split chromosome configuration, and so one of these chromosomes is going to go to the left, and one of them is going to go to the right. If this is the egg, and this is the polar body, then these 2 chromosomes will never go to the next generation. So it turns out that chromosomes, although they can't think, they're not aware of anything, but in an evolutionary sense, they want to be in this egg, and they don't want to be here. So any DNA sequence or any characteristic of these chromosomes that increases the tendency of coming into the egg will get selected for. And anything that increases the tendency of going to the polar body will get selected against. Normally, both chromosomes are pretty identical, so they can't tell which way they're going, and the cell also can't tell which chromosome is which. So there's no chance for a chromosome to cheat and end up in the egg. But if there's a large gross difference between chromosomes, such as one of them being split and the other one not, then that is a little hook for them to get to try to cheat evolution and end up in the egg, regardless of their effect on fitness. Right. So that's what I should have shown. So yeah, the viable products, the products that will go to the next generation, are different in male and female meiosis. Which means that if you start meiosis with 2 homologs distributed among those 4 sperm. The homologs will be distributed equally among those 4 sperm, but only one of the 4 original DNA strands is going to be in the female gamete. So if you have large numbers of gametes, right? If you're thinking of thousands of sperm and thousands of eggs over many generations, then Mendel's laws are still holding up. There's still independent assortment of these individual chromosomes. So each chromosome has a certain allele of a certain gene, and in sperm, Mendel's law is just direct because the 4 products of meiosis are all viable. And in the case of eggs, Mendel's laws are holding in an indirect sense, although each meiotic division only gives rise to 1 allele, so that's not independent assortment. But if you average it over many, many eggs, there should be independence because each egg doesn't know what the other egg is doing. Okay, but if you have some bias such that the red chromosome has an advantage and the red chromosome always comes into the egg through some mechanism, or at least has a higher chance of getting into the egg, then in male meiosis, of course, everything is still the same. But you might end up with a situation like this, where if you look at all the eggs something, let's say a bird or whatever, lays over its lifetime, it will lay an excess of red chromosome eggs over yellow chromosome eggs. Okay, so this is all kind of theoretical. I just said, all right, if there's some difference between the two cells and the chromosome has a way of discriminating that, then you would end up with more of those chromosomes. But does that actually happen in real life? There is strong evidence that yes, it does happen. And it happens in the very case that I was just talking about with primate evolution and the fusion of chromosome 2. So the breakage or fusion of a chromosome at the centromere is what's known as a Robertsonian translocation, because this guy named Roberts found it. And it's one of the most common translocations you can get, one of the most common chromosome breakage or fusion events you can get. The reason it's common is that the fusion of these two chromosomes at their centromeres doesn't lead to a dicentric chromosome. It just leads to a chromosome with a single centromere. So that doesn't lead to any decrease in viability. Before I mentioned that there are some chromosomes where the centromere is actually at the very end of the chromosome, right next to the telomere. So it's not, even though it's called centromere, it's not in the center of this chromosome, it's actually at the end. And likewise, if you break a chromosome directly at the centromere, then both of the breakage products will still have a functional centromere and will still be able to segregate. So there should be no problem in a body cell, a somatic cell, if you break a chromosome in half. Everything will still segregate normally in mitosis. But in meiosis, now you have a little difference. You've got, in this case, let's say this is a simple organism where n equals 3. What you've done is created an unequal number of centromeres on either side of the spindle. Right? So you have 3 chromosomes on one side, so there's 3 centromeres. But since one chromosome broke, now you have 4 centromeres on the other side. And so this is a case where there's actually a difference now in the two. There's a difference in the eggs, and now there's also a difference in something else, which is the number of microtubules that are pulling chromosomes toward one side or the other. So there's also a difference in the machinery that's pulling chromosomes apart. And when you have such a difference, there's this thing called the unequal centromere number rule. If you have an unequal number of centromeres on both sides of the spindle, then you can have this meiotic drive thing occurring. So an example of chromosomes that have a centromere on one end is actually most mouse species have the centromere right here next to the telomere. So they can, that's a lot of work. But those mouse chromosomes can easily fuse together and create a chromosome with one centromere in the middle. All right, but back to here. So we have this unequal centromere number rule, where if you have a chromosome breakage event, then in meiosis 1 in the female, you will have a different number of centromeres on one side versus the other. So what happens when we actually look at humans or mice or chickens, because this is what they did the experiment in, when you actually have such broken chromosomes. Okay, so here they're looking at fusion of chromosomes that are normally aquacentric. So in other words, they took mice that had chromosomes like this, but they found a rare mouse where these 2 chromosomes fused together, and then just looked at the offspring of those mice and asked how many of them have this fused chromosome. So this is the new chromosome. So this would just be like the ape chromosomes fusing together into chromosome 2. How many of them have this new chromosome, and how many of them still have the unfused chromosomes? So first of all, let's look at males. So in males, it turns out that it's just about equal. So you look at 7,000 offspring of these male mice that are heterozygous for this Robertsonian fusion. It's just about exactly half. So half of the male's offspring have a single fused chromosome, and half the offspring have the original unfused chromosome. And that's the same in humans and chickens as well. So in other words, if you are a male and you have this fusion event that happens, your offspring are completely identical and you have completely Mendelian segregation. So it's just like you're heterozygous for a gene and it's segregated equally to your offspring. But of course, the interesting thing is if you look at female meiosis, now it's different. Okay? If you look at the numbers of offspring based in mice, for instance, so remember the original parental condition is this unfused acrocentric chromosomes, and this is the derived condition where they fuse together. If you look at similar number of offspring, about 7,000 offspring, hugely more, there's a huge bias towards keeping the original chromosomes and a bias against keeping the fused chromosomes. But interestingly, if you look at humans and chickens, we also have this bias. We also have a bias when it comes to unequal centromere numbers, but it's the opposite way from mice. So in humans, we want to have this fused chromosome in our going to the eggs, and we kind of reject the two centromere situation. And chickens have the same thing. So that's interesting to begin with. So that was a pretty interesting observation. I don't know exactly which chromosome this was, but it must have, in the human case, it must have been one of the smaller chromosomes that can fuse in this Robertsonian manner. Because some of the small chromosomes are pretty acrocentric. They have the centromere at the end. But that was one case. They wanted to confirm this, so they have another case where you have an unequal number of centromeres on either side, and that is mice that are XO females. So these are mice, normally of course females are XX, but if you had a non-disjunction and you only have one X chromosome, they're still females, and you can still look at how they do their meiosis. And right, it turns out that the number of times that single X chromosome goes into the egg is higher than the number of times the egg doesn't get an X chromosome. So, right, in that case, you're just dealing with, so we have this meiotic spindle and just one X chromosome. And it is more often getting pulled toward the big egg than it is toward the small probe body. So in other words, mouse eggs, in this case as well, they also like to grab as many centromeres as they can. And then the last case was another confirmation. This, instead of looking at a fusion event of two chromosomes, now they're looking at a fission event where you can break two chromosomes apart. So these chickens are heterozygous for, they have one completely intact chromosome and then another that's been broken in half, and that's in the same cell. And then when you look in meiosis, you can see that there's a selection towards having, again, a fewer number of centromeres. So if you look at cases where they had non-disjunction, they're more likely to have a single chromosome going to the egg, either this one or this one, much more likely than they are to have two chromosomes, so one full chromosome and one broken chromosome. And that's the case where you have that's the case when you have partial non-disjunction, and it's also the case where you have total non-disjunction. So just by random chance, you should expect the number of times you have all three chromosomes going to the egg versus 0 chromosomes going to the egg. Those should be the same, just by random chance. But in fact, it's 13 times more likely that none of those chromosomes will go to the egg. So in other words, mice eggs seem to want to have as many centromeres as possible, whereas human and chicken eggs want to have as few centromeres as possible. That seems to be the rule. And then the last piece of evidence that nails this down, that I think is one of the most kind of coolest results in evolutionary biology to come out in the past 20 years or so, is you can get, so these are all kind of experiments where people looked at they knew parents' karyotypes, they looked at offspring karyotypes, and they made some conclusion based on what they saw. But people were already kind of thinking along those lines just based on looking at karyotypes of organisms. Because going back to what the mouse chromosomes look like. So I said, of course, mice eggs like to take as many centromeres as they can. And if you look at it, all these mouse chromosomes are acrocentric. So that they all resemble the case that mice eggs seem to prefer, right? So mouse eggs have this bias. Whenever there's a splitting event, they have this bias towards inheriting 2 versus 1. So that means they have this bias where they want to inherit these acrocentric chromosomes into their eggs. And if you look at their chromosomes, well, they're all acrocentric right now. The implication is that over evolutionary time, as chromosomes went through these Robertsonian splittage events, those were all selected for based on this mechanism of the eggs preferring to have more chromosomes. And of course, if you look at human and also chicken chromosomes, most of our chromosomes look like this instead, with the centromere in the middle. So the idea is that maybe this tendency on a cell division, on a single cell division basis, may be reflected in the actual chromosome makeup of organisms today. And that led to a pretty cool prediction. So if you take the number of chromosomes that a typical mammal has, and then ask what, if it were completely random, so if a given chromosome could either have a centromere in the middle or have a centromere at one end, if that choice were truly random, then you would expect a distribution that looks something like this, where the x-axis here is the percent of acrocentric chromosomes, and the y-axis is the frequency or the number of species that should have that many. So these guys, these evolutionary biologists, looked at all mammal species and just counted how many chromosomes look like this with the centromere at one end versus how many chromosomes look like this with centromeres in the middle. So this one is acrocentric. And this one's metacentric. So if it were random, if it were completely random, then for instance, humans with our 23 chromosomes, you would just expect maybe 11 or 12 of them would look like this, and the other 12 or 11 of them would look like that, because it's random. It's like flipping 23 coins. You would expect about half heads and half tails. So this would be the expected distribution of species if that choice were random. But when people actually went through this and using old texts like the one I showed you, where the guys were just counting the number of chromosomes over and over and over again, what they found was actually a complete opposite. So the black bars here are the actual frequencies of species that have this percentage of acrocentric chromosomes. So humans, I think, are somewhere like this bar here, because we have 1 or 2 acrocentric chromosomes. Most species have either no acrocentric chromosomes. So in other words, most mammal species, the chromosomes are all like this. But then the second biggest group of mammal species are like mice, where the chromosomes are all like that. And species that have 50-50 are extremely rare. And they're probably actually heading one way or the other, because this, as they showed, this tendency of wanting more or fewer centromeres can actually flip over time at some point. But anyway, yeah, this measurement of karyotypes showed kind of an evolutionary reflection of that experimental segregation data. So it kind of beautifully matches up that there is this definite tendency that the egg versus the polar body has this preference And if we think about the geometry of what's going on, we can kind of think about how this kind of mechanism could work. Because the spindle here, it's the environment that it sees over here is slightly different than the environment that it sees over there. And, well, I think the mechanism for how this works is still being worked out. But if you have a spindle that prefers, if you have an egg that prefers a fewer number of chromosomes, then it's thought that this side of the spindle is stronger and has a higher tendency to attract centromeres. And so actually this spindle before cell division is sitting over here, not here in the middle. But this is what it would look like if the higher number of centromeres goes to the polar body, and then the oocyte version would be reversed like this. Okay. So this explains. So this would be, for instance, the mouse situation, where if you have this splittage, that tends to go to the oocyte. So that also kind of agrees, or could help explain why, when there was this rare fusion event between these two separate chromosomes, that reduced the number of centromeres on one side of the oocyte, and so those two fused chromosomes then could become human chromosome 2, because our eggs prefer to have that fewer number of centromeres. Okay, that's it, right? Yeah, this is just a distribution of which clades, so which kind of animals have which kind of distributions. What am I showing here? So this, so this Scadentia, I think, are the shrews, and they tend to have. They are kind of a close group to us as primates. So, there's kind of this phylogenetic tree. So the left side is like humans, where we like to have the fewer number of centromeres. The right side would be like mice. They like to have more number of centromeres. And this is just showing that between somewhere along this line, leading to the split between primates and these kind of shrews, there was a split in which way that balance went. So it's not something that's set in stone. In fact, these guys who studied mice to find this data. These same guys who studied mice, I don't have data to show you, but it's just kind of the story. They found a mouse species on some island in the Mediterranean, where instead of the chromosomes looking like this, they had human-like chromosomes with centromeres in the middle. And then they did a very similar experiment on those mice and saw a more human-like bias in their inheritance. So the idea is that after some time, after a long time of having chromosomes like this, eventually maybe the chromosomes get too short and it becomes a liability to have this many split chromosomes. And so at some point, this mechanism, this preference, will flip, and then all the chromosomes will start fusing again. And that's what happened on this European island. They estimate sometime in the last 1,000 years, so it can go very, very quickly. Of course, mice have a very short generation time. But this force for evolution of chromosomes is something that can change really rapidly, and it's one explanation why the number of chromosomes in organisms can be so variable, and you can't really predict anything. It happens to be the case that humans and our relatives have very similar numbers of chromosomes, but if, for instance, orangutans had something like 10 chromosomes, then we could start to understand why that might be, because maybe a lot of their chromosomes started fusing. Okay, that's it for today. So for those who weren't here, this is going to be what we talk about for the remaining classes. Next week will be sex chromosomes. Then there's a quiz in 2 weeks on everything we've had up until today. And then a few more things, and then the test. Oh, it's still 6 weeks away. Okay, that's it. So, any questions now or after or what?